The overall goal of this procedure is to explore mid-range non-randomness, also called inhomogeneity, of a user-specified genomic sequence. This is accomplished by first examining the oligonucleotide composition of an input sequence and generating a frequency table of oligonucleotides comprising the sequence. This goal is achieved by invoking the SRI analyzer program. The second step is to produce random sequences that have exactly the same oligonucleotide composition as the input sequence. This is achieved by invoking the SRI generator program. The third step of the procedure is to find segments within the input and randomized sequences that are significantly enriched by a particular nucleotide or nucleotide combination, for example GC or AG. This goal is achieved by the MRI analyzer program. The final step of the procedure is to download the files containing sequences of all MRI regions detected by the program. Ultimately, results can be obtained that show a majority of genomic regions of multicellular eukaryotes are significantly enriched by segments containing base compositional extremes detected for each of 4 nucleotides or any of their combinations. These inhomogeneic regions are associated with unusual DNA conformations and/or particular DNA properties.
This method can help answer key questions in the genomics field, such as finding potentially functional DNA elements within vast areas of non-coding sequences, including intergenic regions or introns.
Though this method can provide insights into mammalian genomes, it can also be applied to other organisms such as